Hello viewers, myself Dr. Latesh, working as Assistant Professor of Home Science in Government PG College for Women, ROHTAK. In this presentation, we will discuss about various methods of food preservation. First of all, we must know what is food preservation. We can define it as it is the process of handling or treating the food to stop or slow down the food spoilage. Preservation generally prevents the growth of microorganisms. In this presentation, we will discuss 4 subheadings of methods of food preservation. Number 1, preservation using high temperature. Number 2, preservation using low temperature. Number 3, irradiation. Number 4, dehydration or drying. These are 3 principles of food preservation. Number 1, prevention or delay of microbial decomposition. In this principle, we can follow by keeping out the microorganisms. This is called as asepsis, or by the removal of microorganisms such as filtration, by hindering the growth and activity of microorganisms by low temperature, drying, anaerobic conditions or chemicals. The fourth one, by killing the microorganism through heat or radiation. The second principle is prevention or delay of self-decompression, which can be performed by destruction or inactivation of food enzymes by the process of blanching, or by prevention or delay of purely chemical reactions such as prevention of oxidation by the means of an antioxidants. The last one, prevention of damage because of insects, animals or mechanical causes. First one, food preservation using high temperature. There are some factors that influence the heat treatment during preservation of food. The first factor is nature of food, means composition of food, or we can say amount of carbohydrate, proteins and lipid present in the food, or solute content, moisture content, pH or antimicrobial contents also affect the heat treatment. Generally, carbohydrate, protein, lipid provide the protection to microorganisms against heat. Microorganisms in liquid food and food containing small size particles suspended in liquid are more susceptible to heat. Second one, nature of process. Microbial destruction in the food is expressed in the terms of its exposure to specific temperature for a longer period of time. They are inversely related. As the temperature increases, the time will decrease. We can say low temperature and longer will be the time. Liquid food is heated more rapidly than the solid food. The type of container used for during preservation will also affect the heat treatment. Small container heated more rapidly than the larger containers. The last one, nature of microorganisms. That means which microorganisms are present in food, may be bacteria, yeast or mold. So far, destroying or inactivate these microorganisms, temperature will vary. High temperature food preservation can be done by 3 methods. First one, pasteurization. Second one, canning. Third one, bottling. Then what is pasteurization? It is the process that kills the bacteria in liquid food, generally moderately high temperature that ranges from 62 to 100 degrees centigrade for 15 to 20 minutes. Is used to inactivate the certain enzymes and kill the microorganisms especially present in milk. Since all the microorganisms are not killed, so needs refrigeration after exposure to air. This method was invented by Louis Pasteur in 19th century. Now, what is pasteurization? There are three methods by which pasteurization can be done. First one, high temperature short time method. High temperature short time method employs comparatively high temperature for shorter duration of time, that is 71.7 degrees centigrade for 15 seconds. And the second one, holding method. In this method 62.8 degrees centigrade temperature is employed for 30 minutes. And the last one is ultra high pasteurization. In this type of pasteurization, the temperature is 137.8 degrees centigrade, and that is for 2 seconds. And there are two types of heating. Number one, heating at about 100 degrees centigrade. Formerly, home canners processed all the foods varying length of time at 100 degree or less. This heat treatment was sufficient to kill the every microorganism, but bacterial spores in the food are often was sufficient to preserve even low and medium acidic foods. Many acidic foods can be processed at 100 degree or less. During baking, the internal temperature of bread, cake or other bacteria or other baking products approach but never reaches to 100 degree as long as moisture is present. Heating above 100 degree centigrade, temperature above 100 degree centigrade usually are obtained by means of steam under pressure. In steam pressure sterilizers restarts when liquid foods are to be sterilized before their introduction into sterilized. Cans high steam pressure are used to apply a high temperature for a few seconds. And now, what is canning? Process of applying heat to food that is sealed in containers to destroy the microorganisms that can cause food spoilage. Proper canning techniques are used to kill the microorganism and store the food. Air is driven from container and vacuum is formed in the container as they cool and sealed. Generally, canning is done in the tins. Raw food for canning should be freshly harvested, properly prepared, inspected, graded if desired, through washed before introduction into can. Many vegetables are blanched or scalded briefly by hot water or steam before packaging. Canning is the preservation of food in sealed containers, and generally tin coated steel or glass containers they are used, but increasing use in being made of containers that are partially or wholly of aluminiums or plastic as pouches or solid containers or of a composite of material. Therefore the word canning is generally termed, and now it is replaced by hermetically sealed container. The next process is bottling. Toughened glass is used as heat treatment is done at very high temperature. It is easy to sterilize the glass. Bottle is either filled with raw foods and then heated slowly to boil the foods to destroy the microorganism. Air is expelled from the bottle through a specially designed lid as it cools down and the vacuum is created. Sealed bottle jar prevent the entry of microorganisms as there is no air present in the jar. The next one, food preservation using the low temperature. There are three methods. Number one, chilling or cold storage. Chilling storage is at temperature not far above freezing and usually involves cooling by ice or by mechanical refrigeration. It may be used as main preservative method for foods or for temporary preservation until some other preservative process is applied. Most perishable foods including eggs, dairy products, meats, seafoods, vegetables and fruits may be held in chilling storage for a limited time with little change from their original condition. And this process depends on temperature. The lower the temperature of storage, the greater the cost. Therefore, almost foods will keep best at the temperature just above the freezing point. Next one is relative humidity. The optimal relative humidity of atmosphere in the chilling storage varies with the food stored and with environmental factors such as temperature, composition of atmosphere and the ray treatments. Too low a relative humidity results in loss of moisture and hence of weight, wilting and softening of vegetables and shrinkage of fruits. Too high relative humidity favors the growth of spoilage microorganisms. Highest humidity near the saturation is required for most bacterial growth on the surface of foods. Next one, ventilation. Ventilation or control of air velocities of storage room is important in maintaining a uniform relative humidity throughout the room, removing the odor and preventing the development of stale odor and flavors. The next one is composition of storage atmosphere. The amount and proportion of gases in the storage atmosphere influence the preservation by chilling. Usually no attempt is made to control the composition of atmosphere through store plant foods continue to respire using oxygen and giving off the carbon dioxide. Next one is common or cellar storage. The temperature is common or cellar storage usually is not too much below that of outside air and seldom is lower than the 15 degrees centigrade. Root crops, potatoes, cabbage, celery, apples and similar foods can be stored for limited periods. The deterioration of such fruits and vegetables by their own enzymes and by microorganisms is not prevented but is lower than at atmospheric temperature. Too low a humidity in the storage cellar results in loss of moisture from stored food and too high humidity favors the spoilage by microorganisms. The last one is freezing or frozen storage. Freezing or frozen storage of food in the frozen condition has been an important preservative method for centuries where outdoor freezing temperatures were available. With the development of mechanical refrigeration and quick freezing processes, the frozen food industry has expanded rapidly. Even in the home, the freezing of foods has become extensive now that home deep freezers are readily available under the usual conditions of storage of frozen foods. First one is selection and preparation of food for freezing. Quality of food to be frozen is of prime importance, for the frozen food can be no better than the food was before it was frozen. Fruits and vegetables are selected on the basis of their suitability for freezing and their maturity and are washed, trimmed, cut or otherwise pre-treated as desired. Most vegetables are scalded or blanched and fruits may be packed in the syrup. Meats and seafoods are selected for quality and are handled to be as to minimize the enzymatic and microbial changes. Next one is freezing of foods. The rate of freezing of foods depend on the number of factors such as the method employed, the temperature, circulation of air, refrigerant, size and shape of the package, and kind of food. Three types of freezing available. Sharp freezing usually refers to freezing in air with only natural air circulation or at best with electric fans. The temperature usually minus 23.3 degrees centigrade or lower than, but may vary from minus 15 to minus 29 degrees centigrade. Freezing may take 3 to 72 hours. This sometimes is termed as slow freezing, or we can say quick freezing, in which food is frozen into a relatively shorter time. Quick freezing is variously defined, but in the general implies a freezing time of 30 minutes or less. Usually the freezing of small packages or the units of food. Next one is dehydrofreezing. Dehydrofreezing means certain fruits and vegetables, fish, shrimp, mushrooms now are being frozen by the means of liquid nitrogen. This process is called as dehydrofreezing. Fruits and vegetables have about half their moisture removed before freezing. There are some changes during the preparation for freezing or changes during freezing. The rate and kind of deterioration of food before freezing will depend on the condition of food at the time of harvesting, slaughter, and the method of handling thereafter. The temperature at which the food is held and other environmental conditions will determine the kind of microorganisms to grow and changes to be produced. Freeze drying. Rapid drying system cause outer edge and corner of food dried out, thus fix the shape of food in early stages. The water is removed from the center of food, produces honeycomb structure, got rehydrated when immersed in water, whereas slow drying allows product shrinked and cannot be rehydrated. Water soluble vitamins can be lost during freeze drying. Considering all the drying processes, this method is more effective as the size, shape, color and texture of food remains unchanged. The third method is irradiation. Irradiation. In this method of food preservation, ionizing radiations are applied to food to kill the microorganisms. No risk of radioactive radiation on the food. It is more expensive method. No visible changes are observed other than undesirable factors in the food. Changes in organoleptic properties of foods are not observed in the irradiated foods. Factors that influence the effectiveness. There are three factors that will affect the intensity, time and penetration of rays, intensity of rays, and at what time these rays are given to the food. Kind of ionization radiations. There are various types of radiation. Number one, gamma rays, X-rays, beta rays, cathode rays and electrons. Food irradiation uses reduce the insect infestation in grains, spices, fruits and vegetables, inhibit sprouting such as tubers and bulbs, retard ripening in such fruits, inactivate the parasites in case of meats and fish, eliminate the spoilage microbes in case of fruits and vegetables, extend the shelf life of poultry, meat, fish and shellfish, decontaminate the poultry and meat, sterilize the food and feeds. The last method is drying or dehydration. Drying. Microorganisms cannot grow without moisture. In this process, removal of moisture using warmth or high temperature. Dried foods have more shelf life that can be stored at ambient temperature. There are several commercial methods of drying used in foods: sun drying, freeze drying, tunnel drying, spray drying, etc. There are various processes such as sweating, packaging, and pasteurization. Mainly the drying is of two types. Number 1, natural drying, which is done by sun drying we can say, and second one is mechanical drying by various procedures. These are the differences between sun drying and mechanical drying. Solar drying is limited to climate with hot sun and dry atmosphere to certain fruits like prunes, figs, raisins, apricots, and peaches, fish, rice, and other grains. Mechanical drying, most of the methods. Artificial drying involves passage of heated air with control the relative humidity over the food to be dried, or passage of food such as air through such air. These are the examples of foods on which type of dryers are used for drying. Vegetable confectionery, fruits, pectin. The type of dryer is compartment and tunnel dryer. Grass, grain, vegetables, fruit, nuts, breakfast cereals, conveyor belt. Grass, grain, apple, pomace, lactose, poultry, manure, and starch, rotary dryer. Coffee, milk, tea, fruit, purees, spray drying. Milk, starch, pre-digested infant foods, soups, brewery, and distillery byproducts through film drum. Starch, fruit pulp, distillery waste products, crops, pneumatic, coffee, essence, meat extracts, malted, and other confectionery through freeze dryer and vacuum dryer. There are some question and answers through this presentation. These are multiple choice question answers, and the text given red color are the right answer. The drawback of X-ray in food preservation is both A and B is the right answer. Low efficiency and high cost. Rays are directional and less penetrating. Cathode rays are directional and less penetrating. The simplest dryer, example of simplest dryer is kiln dryer. 99% bacteria are removed by blanching. Temperature in cellar storage is 15 degree centigrade. Chilling storage enable the preservation products for limited period. Rapid slowing of chemical and enzymatic reaction in foods are achieved in quick freezing. The father of pasteurization is Louis Pasteur. Heating of canes can be done by boiling water bath, using steamer, oven heat. The right answer is all of the above. These are some references used in this presentation. You can go through these books. Thank you viewers for listening my presentation patiently.